This is Deborah Zaritsky in Bio 515 at Grand Canyon University. Today is April the 15th. Reporting on the muscular system for Dr. Ott. We had just finished talking about the CNS and the PNS, knowing that the spine and brain make up the CNS and our nerves make up our PNS. We know that these are connected by afferent and efferent neurons, and we're connecting our sensory with our motor in the somatosensory cortex. So as these are connected to our muscle tissue, what exactly is the difference between all of our different types of muscle? So we have skeletal, smooth, and cardiac. I will be mainly reporting on the skeletal and the smooth muscle. We know that skeletal muscle is striated or has these dark bands when looking under the microscope. We're going to see some other components here, mainly the sacromere, Z-line, I-band, and anatomy of that sort in our skeletal muscle. This is going to be connected to our somatic nervous system, where we mainly have some voluntary muscle control with our motor neurons. These skeletal muscles are going to be attached to our bones by connective tissue known as tendons. The smooth muscle is going to be a different shape. It's going to have one nucleus, and it's mainly going to be in our autonomic nervous system, where we have some chemical processes that are going to be controlling this tissue within hollow organs and tubular tissues such as our blood vessels. When we look closer inside of the skeletal muscle, we can see from the macro to the micro where we have our muscle tissue. Inside of the muscle tissue are going to be these linear tissues called myofibrils. About 80% of the volume of a muscle fiber is the myofibril. Inside of the myofibril, we're going to find sections called sacromeres. These sacromeres are divided by these positions called as Z-lines. Z-lines run vertically, whereas these other components called bands are going to run horizontally. Sacromeres are sections of the myofibrils that allow contractile movements with the interaction between specialized proteins called actin and myosin. These proteins are set within structures called filaments. We can see a depiction here. This is kind of a generalized picture. When we look a little bit closer, we're going to see that these proteins are going to be set on these other structures called filaments. There are thin filaments and there are thick filaments. The myosin is going to be known as a thick filament that sits in the center of the sacromere. The thin filaments are going to be running across. In this picture, it kind of shows that these structures, the dark ones, are going to be the thin filaments, whereas the lighter ones are going to be the thick filaments. Thick filaments are stationary. The thin filaments are going to be moving. We can see that actin, one of the proteins, is going to be attached to the thin filament. In a relaxed muscle fiber, contraction is going to rely on something called a cross bridge. The cross bridge is going to be made when the head of the myosin molecule comes across the receptors for the actin. So the actin is going to bind to the heads of the myosin molecule. Actin is a thin filament and myosin is a thick filament, and according to Sherwood, an I band consists of the remaining portion of the thin filaments that do not project into the A band. This is visible, the I band is visible in a dense vertical Z line. The Z line is flat, is a cytoskeletal disc that connects the thin filaments at the adjoining sacromeres. In order for the thin filaments to come together to make a contractile motion, the calcium must be released after there is an excited stimuli. Once the calcium is released, then the myosin and the thin filaments of actin are going to be sliding past the myosin heads. When this happens, the thick filament produces sort of a walking motion. Each action is independent of each other and requires a new release of ATP for every cycle. There are two other proteins by the name of troponin and tropomyosin. These are wound around the thin filament. If calcium is not released, the troponin and the tropomyosin will be blocking the actin receptors. Therefore, contraction is not possible. A cross bridge occurs when the thick and thin filament connect and propel forward. This is also known as a power stroke, which allows the thin filaments to come together. The sliding filament mechanism is what happens when the I band does not overlap with the thick filaments. The thin filaments further overlap the thick filament during the inward slide. Thin filaments pull the Z lines to which they are attached closer together. When contraction does happen, this happens in two different types of skeletal muscle fibers. Even though there are two types, the second type is divided into two other subsections. We call these a slow twitch, type 1, and then we have the fast twitch, which is type 2A and B, or fast twitch 2X. The slow twitch is going to have a slow reaction time with a high resistance to fatigue, uses oxygen. We're going to have a lot of mitochondria that are going to produce a lot of ATP. There's a lot of oxygen, which allows for long contractions. We're going to be storing energy in triglycerides in the form of fat. So these are going to be our large muscles, mainly found in our back and our legs, used for strong forces. When we have a fast twitch muscle, those are just short-term actions. These are due to anaerobic activity. We do not have a lot of mitochondria there, hence there's not a lot of oxygen. These types of fibers are going to be using raw ATP for energy. So an example here is like flicking the finger or shaking a hand. There's something called a tension-length relationship, and this does occur within the skeletal muscle fibers. I have a picture here of a mechanism used in a pinball machine, so that when you pull the mechanism back this way and then you release it, the spring allows for much more force going in this direction. This is showing kinetic energy. So in the tension length relationship, not only do we see some physics, but there is some math there also. So this does have to do with the size of the resting muscle tissue. So some examples that I saw where scientists were looking at the gastrocnemius or the calf muscle of a frog. If they were to disconnect the calf muscle from the tendon area, the muscle would be smaller than how it is attached to the leg bones. So when you increase the length, you increase the tension, much like pulling on a rubber band, and in this case, the pinball mechanism. So when you put more tension on the muscle, then the myosin heads are going to be pulling with more force. So the position of that skeletal muscle is appropriate for the load that it would need to carry in order to do work. And that goes back to our type A or type 1 types of muscle tissue. So according to Sherwood, there are light chain molecules known as necklaces, where the heads of the myosin molecules are near the neck region. And these light chains are going to be tacking on to the actin proteins. So now we're talking about smooth muscle, where there is no troponin or tropomyosin. And again, the myosin will interact with the actin only when this light chain of the myosin is phosphorylated. So these are found in hollow tubes and are such as our digestive system and our blood vessels. So in conclusion, there is a large difference in between our skeletal and smooth muscle tissue, and of course our cardiac as well. In our skeletal muscle, again, we know that it's striated. It's in our somatic nervous system or voluntary nervous system, acting on action potentials or depending on the release of calcium in order to access our actin binding sites. We know that our type 2 muscle fibers may be fast processing, whereas our type 1 is kind of a slow processing. We know that skeletal muscle has a length tension relationship because it's attached to our bones there, whereas our smooth muscle does not have this relationship. We do not have sacromeres within our smooth muscle tissue. We know that they are multi-unit or single unit, and they are going to be either phasic or tonic. And we know that these are slow processing, such as in our digestive. But together, they both use thick and thin filaments in their sliding filament mechanisms. There's a presence of actin and myosin. Our nervous system initiates the contraction. We have different nerve fibers for both our skeletal and our smooth. And there is a rise in our calcium levels. Here are my references. And thank you.